subtle fields in our environment.
Okay, good. Well, let's dive into the blood cell analysis work that you've done, the live blood cell analysis. And we're going to show those two slides on the screen here, Beverly. So talk us through, what are we looking at here, and how are these effects caused?
Well, first, let me say what the design of this study. So this study had 10 subjects ranging in age from 27 to 75 years old. And first, they had to have very good-looking blood. So they actually were eating a very, very good diet, because initially, if their blood didn't look good, I couldn't use them in the study. And most of them were cell phone users, about 80%. I did the study about 5 years ago. And I first took an initial blood test from the fingertip, a droplet of blood placed on a microscope slide under a dark field microscope. And there you see at the baseline in the upper left-hand corner how the blood looks initially without any exposure to cell phones. In this study, they had to fast and refrain from use of Wi-Fi or cell phones for 4 hours before they came to see me in the study. And then after the baseline test, I presented them with a cell phone, a smartphone running on 4G that was turned on, but they did not make a call. They wore it in a backpack for 45 minutes. The phone was in readiness mode to receive a call or text, but it did not. And then I took another blood test, and in this case, you see there were changes in the blood. As shown in the upper right hand of the photographs, the red blood cells became sticky and stuck together in what we call rouleaux or rolls, like looking at the edge of a roll of nickels. And with this, the peripheral blood circulation is compromised. The blood cells actually have to scrunch down in order to go through the tiniest microcapillaries in the body. And so when the blood is like this, it can't. So microcirculation is compromised. Not only did I look at fingertip blood, because in some cases they were holding the phone later, I looked at blood from the toe as far away as possible and found the same stuck together red blood cells. And after the second condition, which I call the carrying condition, then I had them use the phone for an additional 45 minutes. They went online on the internet, and they also placed the phone near the head for 2 5-minute phone calls. And at the end of 45 minutes, I took a third sample of blood and found even further disturbing changes in the blood, as shown in the bottom photograph. Now the cells were broken apart from the rouleau, but they were distorted. The membranes showed shape changes, and in many cases were spiky, appeared like bottle caps, or spiky like a sea urchin. In fact, in hematology, these spiky cells are called echinocytes, after the echinoderm, the sea urchin. And they're not a healthy sign in hematology. This is a sign of, again, compromised peripheral blood circulation. So these are short-term changes due to, say, a 90-minute exposure to cell phone radiation, but that's a small part of the day, and people are using these cell phones throughout the day, as we know, and also exposed to Wi-Fi and innumerable sources from the environment, which is going to be magnified if 5G is